Hello dear learners, welcome to Crown Biology classes. This is Rohit here and I will be discussing the most important topic of genetics today, that is the chromosomes. So under this topic I will be covering the main topics which are as follows. First one is introduction, second one is comparison of prokaryotic and eukaryotic chromosome, third one is shapes of chromosomes, and the fourth one is structure of chromosomes. So first of all, starting with the introduction part. A chromatin is an organized structure of DNA and protein. Organized structure of DNA and protein, that means with DNA there are present associated proteins with it. And those proteins are named as histones. That is found in the nucleus of eukaryotic cells. So this chromatin is present in which organelle? It is present in nucleus of eukaryotic cells. Eukaryotic cells are those cells in which the nucleus is surrounded by a well-developed nuclear membrane. Chromatin and chromosomes are basically the same thing. The only difference is that chromatin is less condensed and extended DNA, while chromosomes are highly condensed DNA. Something which is less condensed will be having more length. Hence, chromatin is an extended DNA. While chromosomes are highly condensed DNA, that means they will be having lesser length. The chromatin condenses to form chromosomes. This means chromatin is lower order of DNA organization, whereas the chromosomes are higher order of DNA organization. The second point is the word chromosome. It comes from the Greek word chroma. Chroma meaning color and soma meaning body. And it is named as such due to their property of being very strongly stained by particular dyes. A particular set of dyes do stain these chromosomes very strongly, and hence the name chromosome. Now we will discuss about the discoverer and their discoveries. E. Strasburger in 1875 for the very first time discovered thread-like structures. Those thread-like structures were nothing but chromosomes which appeared during cell division, which appeared during cytokinesis when the two cells were dividing. Walter Fleming in 1878 introduced the term chromatin to describe the thread-like material of the nucleus. So for the very first time, Walter Fleming termed those thread-like material as chromatin that became intensely colored after staining. As we know, this is the property of chromosomes or chromatin. The present name chromosome, it was coined by W. Waldeyer. R.D. Kornberg in 1974 proposed the nucleosome model of the basic chromatin material. Nucleosome model, it explains the organization of DNA and the associated proteins, and those associated proteins were histones in the chromosome. It also further explains the exact mechanism of the folding of the DNA in the nucleus, how the DNA is folded and how it is present in the nucleus. And this nucleosome model is the most accepted model of chromatin organization. The term nucleosome, it was coined by P. Audet in 1975. Chromosomes carry the genetic information and so are called the hereditary material of a cell. As the genes, the units of heredity, these are present on chromosomes, and these genes are responsible for the transmission of characteristics from parents to offspring. and hence the chromosomes are called the hereditary material of a cell. The structure of chromosomes differs in prokaryotes and eukaryotes. The prokaryotes, for example bacteria, which do not have a membrane-bound nucleus. That means their nucleus is not surrounded by a well-developed nuclear membrane. They have only one chromosome. Whereas the eukaryotes, the chromosomes occur in pairs, and the chromosomes are known as homologous chromosomes. And the haploid set of the chromosomes is also known as the genome. And we can define genome as the complete set of genes in a cell or a living thing. Now I will discuss about the comparison of prokaryotic and eukaryotic chromosomes. So the first point is the prokaryotic chromosomes, they have simple organization, but the eukaryotic chromosomes, they have much more complex organization. Second point is prokaryotic cell has only one prokaryotic chromosome as we have discussed earlier, and this prokaryotic chromosome is circular in shape. An eukaryotic cell has a number of eukaryotic chromosomes, and those chromosomes differ in shape and size. The third point is the prokaryotic chromosome is made up of a single circular DNA, and that single circular DNA is associated with a few proteins, whereas eukaryotic chromosome has a large amount of DNA, and that DNA is associated with much more number of basic proteins, and those basic proteins are known as histones. Now I will discuss about the shapes of chromosomes. Each chromosome has a condensed or constricted region called the centromere. So there is a specific position on the chromosome which is condensed or constricted. So that specific position is termed as centromere. The position of centromere is specific to a chromosome. That means this position can vary and it can be different in different chromosomes and varies in different chromosomes. The centromere divides the chromosomes into two parts, and those two parts can be equal or unequal, and each one is called an arm of the chromosome. Depending on the position of centromere, chromosomes acquire different shapes. Accordingly, chromosomes have been classified into the following four categories. So the first one is telocentric chromosomes. In telocentric chromosomes, the centromere is present at the proximal end. That means the centromere is present at one end, giving them rod-like shape. The second type of chromosome is acrocentric chromosomes. These are also rod-like in shape, just like telocentric, but the difference is the centromere is present near the extreme end, so one arm is very long and other is very short. The third type is submetacentric chromosomes. These are J or L-shaped chromosomes in which the centromere is slightly displaced from the center. The centromere will not be exactly placed in the center, rather the centromere will be placed slightly away from the center, so both the arms are unequal. And the fourth type is metacentric chromosomes. These are V-shaped as the centromere is present right in the center, so both the arms are equal. As we can see in this picture, in metacentric chromosomes, they are having V-shaped and the centromere is present right in the center, so both these arms are equal. In some metacentric chromosomes, the centromere is slightly displaced from the center. As you can see, the centromere is not present right in the center, rather it is slightly displaced, so both the arms are unequal. The third type is acrocentric chromosomes. These are also rod-like in shape and the centromere is present near the extreme end. So one arm is very long and the other is very short. And the fourth is telocentric chromosomes, in which the centromere is present at the proximal end, giving them rod-like shape. Now I will discuss the structure of chromosomes while describing the structure of the chromosomes during various phases of cell cycle. The various phases of cell cycle are gap 1 phase, synthesis phase, gap 2 phase and mitosis phase. Cell biologists have introduced many terms for their various components. In general, chromosomes are composed of various components. So the first component involved in the formation of structure of chromosome is chromatid. At mitotic metaphase. At metaphase stage of mitosis, each chromosome consists of two symmetrical structures, and those two symmetrical structures are called chromatids. So when we do observe the metaphase stage under microscope, we observe that the chromosome consists of two symmetrical structures known as chromatids. Each chromatid contains a single DNA molecule. So the number of DNA molecule will be equal to the number of chromatids and vice versa. Both chromatids are attached to each other by the centromere. As I have already told you that there is a region of constriction on a chromosome that is known as centromere. Likewise, the chromatids are also attached to each other by the centromere, and they become separated at the beginning of anaphase. So during anaphase stage of mitosis, the sister chromatids they start migrating towards the opposite poles, and hence as a result of this they start separating. The second component involved in the formation of structure of chromosome is chromonymata. During mitotic prophase, during prophase stage of mitosis, the chromosomal material becomes visible as very thin filaments. And when we do observe the chromosomes during the prophase stage of mitosis, the chromosomal material becomes visible as very thin filaments, and those thin filaments are called chromonymata. That is the term coined by Wajdowski. A chromonyma represents a chromatid in the early stages of condensation. So a chromonyma is nothing but a chromatid, and during early stages of condensation we do call that chromatid as chromonyma. Therefore, chromatid and chromonyma are two names for the same structure, and that structure is a single linear DNA molecule with its associated proteins. These proteins are always associated with the DNA so as to make it stable. The chromonymata form the gene-bearing portions of the chromosomes. Chromonymata is the particular region on the chromosome which bear the genes. The third component involved in the formation of structure of chromosome is chromomeres. The chromomeres are bead-like accumulations of the chromatin material that are sometimes visible along interphase chromosomes. So during interphase stage of mitosis, we do observe the bead-like accumulation, and those bead-like accumulations are nothing but the chromatin material. The chromomere bearing chromatin has an appearance of necklace in which several beads occur on a string. So the chromatin material it is present like beads on a string, and therefore it gives an appearance of a necklace. The fourth component involved in the formation of structure of chromosome is centromere and kinetochore. Centromere is the region of the chromosome to which are attached the fibers of mitotic spindle. Mitotic spindle is a structure that forms during cell division, and the main function of this mitotic spindle is to separate duplicated chromosomes. The centromere lies within a thinner segment of chromosome, the primary constriction. So there is a specific position on a chromosome where the centromere is present, and that position is known as primary constriction. Centromeres are found to contain specific DNA sequences with special proteins bound to them, forming a disc-shaped structure that is called kinetochore. So this kinetochore is nothing but a centromere that contains specific DNA sequences and special proteins. So this kinetochore is a complex of proteins associated with the centromere of a chromosome during cell division, to which the macrotubules of the spindles are attached. Under the electron microscope, the kinetochore appears as a plate or cup-like disc situated upon the primary constriction or centromere. This kinetochore is closely associated with the centromere, and when we do observe this kinetochore under the electron microscope, it appears as a plate or cup-like disc. The main function of the kinetochore is to provide a center of assembly for microtubules. That is, it serves as a nucleation center for the polymerization of tubulin proteins into microtubules. First of all, this kinetochore, it plays a key role throughout mitosis as it mediate direct attachments between microtubules and centromeric DNA. And the function of this kinetochore, main function is to provide a center of assembly for microtubules. These microtubules are the major components of the cytoskeleton. And the alpha and beta tubulin subunits, these are assembled into linear protofilaments. And hence, it serves as the nucleation center for the polymerization of tubulin proteins into microtubules. The fifth component involved in the formation of structure of chromosome is telomere. Each extremity of the chromosome has a polarity and therefore it prevents other chromosomal segments to be fused with it. Each end of the chromosome is having a polarity, and due to this polarity, the other chromosomal segments cannot fuse with it. The chromosomal ends are known as the telomeres. So telomeres are the ends of the chromosome which possess polarity. If a chromosome breaks, the broken ends can fuse with each other due to the lack of telomeres. Only due to the lack of telomeres, the broken ends can fuse. Otherwise, the chromosomal ends cannot fuse. The sixth component involved in the formation of structure of chromosome is secondary constriction. The chromosomes, besides having the primary constriction, or that is also known as centromere, they possess secondary constriction at any point of the chromosomes. So this secondary constriction, it is constant in position. That means its position is specific on a chromosome and its amount is also fixed. That means its extent. These constrictions are useful in identifying particular chromosomes in a set, and due to this constant nature, these constrictions are useful in identifying particular chromosomes in a set. The seventh component involved in the formation of structure of chromosome is nucleolar organizers. These areas are certain secondary constrictions, and these secondary constrictions contain the genes coding for 5.8S, 18S and 28S ribosomal RNA. And when these genes code for these ribosomal RNAs, it will induce the formation of nucleoli, and hence the name nucleolar organizers. The secondary constriction may arise because the rRNA genes are transcribed very actively. So the rRNA genes that are coding for 5.8S, 18S and 28S ribosomal RNA, when they are transcribed very actively, then this secondary constriction arises and thus interfering with the chromosomal condensation. In human beings, the nucleolar organizers are located in the secondary constrictions. So in human beings, their position is specific. And these nucleolar organizers, they are located in the secondary constrictions of which chromosomes? Chromosomes 13, 14, 15, 21 and 22. And all of these chromosomes are acrocentric. That means the centromere is present at the extreme end. So one arm is very long and the other is very short. And these chromosomes do have satellites. This is the next component we are going to discuss. The eighth component involved in the formation of structure of chromosome is satellite. The chromosomes bear rounded, elongated or knob-like appendages. So these rounded, elongated or knob-like appendages are known as satellites. The chromosomes with the satellites are designated as the sat chromosomes. This is a schematic diagrammatic representation of chromosome. The components of chromosome, which I have discussed just now, are very well explained by this beautiful diagram. The outermost covering of the chromosome is called pellicle, and this pellicle is very thin and it is formed of achromatic substance. And this pellicle, this membrane encloses a jelly-like substance which is usually called matrix. So this matrix is a jelly-like substance, and in the matrix is present the chromonema or the chromonemata. The matrix is also formed of achromatic or non-genic material. These are the bead-like accumulations present on a string, and these are giving an appearance of a necklace. So these are known as chromomeres. So this is a sad chromosome, that means in which an elongated knob-like appendage is present, that is known as satellite. Kinetochore, these are the proteinaceous discs, and these discs are closely associated with the centromere, that is also known as the primary constriction. Telomere, the chromosomal ends are known as the telomeres. So this is showing telomere. In the second diagram, we can see the centromere is the place where the sister chromatids are attached. These are the sister chromatids. And this is the centromere. And the centromere is closely associated with the kinetochore. So during metaphase, the kinetochore microtubules, these are the kinetochore microtubules, they pull the sister chromatids back and forth until these sister chromatids align along the equator of the cell called the equatorial plate. So this is the characteristic feature of the metaphase chromosomes. Ending this video on a positive note, this is an inspirational quotation which says, "Everyone is unique because no one is the same." If you like this video, then please do like, share and subscribe, and don't forget to press the bell icon so that whenever I do upload a new video, you may get notified. Thank you.